Today we'll study monohybrid cross. Now, how will you define monohybrid cross? It can be defined as a cross between parents differing in one heritable trait or one pair of contrasting character. The word mono means single or one. That means in monohybrid cross, only one character can be studied at a time. As we know, Mendel studied 7 characters of pea plant, so we can choose any one character for monohybrid cross. These are few of the characters that were studied by Mendel. Height, this is the character of plant and its traits are tall and dwarf. The next character is color of seed and its traits are yellow and green. Next one is position of flower and its traits are axial and terminal. So one thing you have to remember is whenever we are doing a cross, we must know which trait is dominant and which is recessive. So in case of height, tall is dominant whereas dwarf is a recessive trait. Whereas in color of seed, yellow color of seed is dominant over green color of seed. In position of flower, axial position of flower is dominant while terminal position of flower is recessive. So all these are dominant traits and all these are recessive traits. Because until and unless we know which is dominant and which is recessive, we won't be able to perform a cross. Now we'll see monohybrid cross with the help of an example. In the textbook, the example that is given is plant height. Hence, we have taken another example, that is shape of seed. Mendel has studied 7 characters. Out of 7 characters, we can choose any one of the character for monohybrid cross. Because previously also I have told you the word mono means only one character. So this is one of the 7 characters that we have chosen as an example for monohybrid cross. This is a character, but what are its traits? The shape of seed, it has got 2 traits. One of it is a round shape of seed and the other trait is a wrinkled shape of seed. So these are the parents that will be used for the monohybrid cross. So first, whenever we are starting a cross, we need parents, that is the cross will begin with parental generation. Once it starts with parental generation, then we'll get F1 generation, then again second parental generation and then only we'll get F2 generation. Now to start with the cross, we'll take the parents first and we'll cross the parents. So the first parent is, that is male parent, it is with a round shape of seed. So this is the first parent, that is male parent. It is crossed with another parent. I am using different color for that. The other parent, that is female parent, it has wrinkled shape of seed. So these are the parents that are crossed. One of the plant, it is having a round shape of seed which is crossed with another pea plant which is having a wrinkled shape of seed. A round and wrinkled, these are the phenotypes of the parents. So these are phenotypes. Then we have to mention the genotype of the parent. So as we have used red color for round shape of seed, to mention their genotype, we'll use the same color. So these are the genotypes for round shape of seed. Similarly, we have to use the same color that we have used to mention phenotype of the seed. these are the genotypes and these are the phenotypes. Phenotype is related to the external appearance of the individual for a particular trait. That means we can see the shape of the seed, but we can't see their genotypes. So this is the genetic constitution, hence it is called as genotype. Now these are the phenotypes, these are the genotypes. The next step will be their gamete formation. For gamete formation, one thing you have to do is you have to mark an arrow here to show that these are the gametes. So this arrow is important. Now the gamete that will be formed from male parent is, this is the male gamete that is formed. In female parent also will get another gamete and this is the female gamete. So from male parent we have got male gamete, from female parent we have got female gamete. Once the gametes are formed, the next process is the process of fertilization. So for fertilization, we'll have to show slanting arrows like this. So this slanting line means this is the process of fertilization that is taking place. Fertilization means fusion of male gamete with female gamete is called as fertilization. So after fertilization, we will get like this. So now we have got the first generation, which is called as F1 generation. What we have done for the cross, monohybrid cross, first we have crossed the parents differing in their contrasting characters. One of the plant is having round shape of seed, other plant is having wrinkled shape of seed. This is their genotype. After that we got the gametes. Once the gametes were formed, fertilization, and then the next generation, which is called as F1 generation. So this is the genotype for F1 generation. What will be its phenotype? The phenotype for this will be, out of two parents, one of it will be expressed. So capital R and small r, it will be round shape of seed. Now one thing that you have to remember is, whenever we are using the genotypes, for round we have taken R and R, but for wrinkled we can't use W as the alphabet. So this you have to keep in your mind. Whenever you are using the genotypes, at that time you just have to use capital alphabets for dominant one and small alphabets for the recessive parent. As we have already studied, round shape of seed, it is dominant over wrinkled shape of seed. So for dominant parent we have used capital letters, for wrinkled parents we have used small letters. Now since both are same, capital R, these two are also same, small r, that means these are homozygous. But you can clearly see for the first generation, that is F1 generation, one is capital R whereas the other one is small r. This means that it is heterozygous since both are different, or we can also call it as hybrid. But the phenotype for this offspring in F1 generation is round shape of seed. Now again, one thing you have to remember, that the characters which are expressed in F1 generation are called as dominant, and this is the first law of Mendel called as law of dominance. So simultaneously, along with monohybrid cross, we will be studying laws also. Two laws will be studying. The first one was law of dominance, which is expressed in the F1 generation. What it says, the alleles or the traits that express in F1 generation are called as dominant. after F1 generation, the next step that we have to do is we have to cross this with itself only, and it is called as selfing of F1 generation. Now this statement is very important in the diagrammatic representation. So we have to mention the statement as selfing of F1 generation. Now this will form parents for second generation. These were the parents for first generation, hence it is called as first parental generation, and now they will be called as second parental generation. Now the phenotype for second parental generation is the male pea plant, it has round shaped seeds, whereas female pea plant, it has also round shaped seeds. That means in the first parental generation, both the parents were different. One was having round shape and the other parent was having wrinkled shaped seeds. But for the next generation, both the parents are having round shaped seeds only. So this was the phenotype. Now their genotype will be, the genotype for male parent, it will be capital R and small r. This is genotype for male parent. Now genotype for female parent will also be same, that is capital R and small r. So these were the phenotypes, that is round shape of seed, and their genotype will be capital R small r for male parent, capital R small r for female parent also. Now the next step will be gamete formation. Now male parent, it will form gametes, and the gametes will be, they will separate from each other. So this is one gamete, this is second gamete. Now for female parent, for this also, this is one gamete and this is another gamete. Now these are the gametes for second generation. Now one thing you have to notice, or one thing you have to remember is, since this is heterozygous, two different type of gametes will be formed. In this case, both the parents were homozygous, so the gametes formed were also of same type. But here the parents are heterozygous, hence the gametes formed are different types of gametes. Now this states that here we have to study second law, that is law of segregation or law of separation. According to this law, the gametes, they are separated from each other, and that is also called as segregation. And this separation is very important because both of them, they will express truly for their character. Hence this law is also called as law of purity of gametes. In F1 generation, we have seen the first law, that is law of dominance, and here when the gametes get separated, it states that second law, that is law of segregation or law of separation, which is very important because if it does not happen, then recessive characters will not express in the second generation also. Because in the first generation we have seen only dominant characters were expressed, whereas for second generation, dominant as well as recessive, both the characters will be expressed separately. Hence the law of segregation. Now once the gametes are formed, the next step is we have to place the gametes in the checkerboard. So this is Punnett square or checkerboard, which is required to predict the progeny for F2 generation. Now we'll place the gametes in this, but for that, in the corner of the checkerboard, we have to mention the parents. So first we'll mention the parents, and for that symbols are used. So this is for male parent, and this is the next symbol which is used for female parent. So this is male parent and this is female parent. Separately we'll place the gametes. This is capital R. This will also be capital R. and this is small r. Similarly here it will also be small r. Now on this side of checkerboard we have written gametes of male parent, and on this side of checkerboard we have written gametes of female parents. Means both the parents' gametes are written in the checkerboard. Now the next step is we have to predict the progeny now. For that what we have to do? We have to cross this R with this R. So the result will be, it will be like this. R crossed with R. You don't have to write R square. Instead of that what you have to write is, you just have to write the alphabets next to each other. This is one capital R and this is another capital R. Now we have to cross this capital R with the small r. You don't have to calculate or you don't have to multiply it. You just have to write the alphabets side by side. Now comes the next one. The small r has come first. In this case, what we have to do is, instead of writing the small r first, we'll be writing the capital R and then only we'll be writing the small r. So now we have to cross small r with the capital R. So for this, first we'll be writing this capital R, and now we'll be writing this small r. Now comes the last one. We have to cross this small r with this small r. So simply we'll be writing the two small r's next to each other. So now our checkerboard is completed. On the basis of this, we have to calculate the phenotypic ratio and the genotypic ratio. Now the phenotypic word, it is related to the phenotypic characters, that means the external appearance of the individual, how it appears. So out of 4, the 3 plants, 1, 2 and 3, they show round shape of seeds. That means for phenotypic ratio, for the round shape of seeds, since they are 3 in number, we'll write the number 3. So these are 3 round shaped seeds, pea plant, and there is only one plant which is having wrinkled shaped seed. So for that we'll write the number 1 because there is only one plant. So out of 4, 3 plants are having round shaped seeds, hence the number 3. Only one plant is having wrinkled shaped seeds, hence the number 1. That means the phenotypic ratio for monohybrid cross is 3:1. Similarly, we'll calculate the genotypic ratio. This genotypic ratio also we have to calculate from the checkerboard itself. So now we'll see the numbers or the ratio. For that, we have to see the genotypic or the genetic constitution of the individual. And these alphabets, they represents the genetic constitution. So we just have to observe the capital alphabets and the small alphabets or heterozygous or combination of both capital and small. How many times they are repeated in the checkerboard. So first we'll see capital R, capital R and capital R. How many times it has occurred in the table? Only once it has occurred. So this is the place where both the R are capital. For this we'll write the number 1. So this is the first genetic constitution. Now we'll see for the second one. How many times capital R and small r is repeated in the checkerboard. So this is 1 and this is second one. That means in the checkerboard it has occurred twice. So for this we'll write the number 2. So 1 as 2, 2 it has become. And now the last one, small r and small r, it has occurred only once in the table. So for this also we'll write the number 1. That means phenotypic ratio for monohybrid cross is 3:1. 3 refers to round shape of seeds. hence the number 3. This 1, it represents the wrinkled shape of seed, hence the number 1. And the phenotypic ratio becomes 3:1. For genotypic ratio, capital R only once, hence the number 1. Capital R, it has occurred twice, hence the number 2. Small r has occurred once, hence the number 1. So these are the phenotypic and genotypic ratios for monohybrid cross. So one thing you have to remember, in F1 generation only dominant characters will be expressed, whereas in F2 generation both dominant as well as recessive characters will also be expressed. It is very easy. The first half, it consists of phenotype, genotype, gametes and then F1 generation. Similarly, for second half, again phenotype, genotype, gametes and checkerboard as well as the ratios. So this is how the cross takes place, and this is all about monohybrid cross. Thank you.